Hello, and welcome students. Today's our topic of discussion is difference between gram-positive and gram-negative bacteria are unicellular organisms that are found everywhere, from soil to water to our own. They can be classified in many different ways, but one of the most important distinctions is between gram-positive and gram-negative. This classification is based on the way that these bacteria react to a particular staining technique called the gram. The gram staining technique was developed in the late 1800s by a Danish bacteriologist named Hans Christian Gram. The stain involves using a crystal violet solution followed by a solution of iodine, and then washing with alcohol or acetone, and then a counter stain such as saffron. The resulting color helps to distinguish between different types of bacteria. Gram-positive bacteria will appear purple or blue under the microscope, while gram-negative bacteria will appear red or pink. The main difference between gram-positive and gram-negative bacteria is the structure of their cell wall. Gram-positive bacteria have a thick layer of peptidoglycan in their cell walls, which retains the crystal violet iodine complex and gives them their purple. In addition, they have teichoic acid, which is a type of molecule that helps to maintain the structural integrity of the cell wall. Gram-negative bacteria, on the other hand, have a thinner layer of peptidoglycan in their cell walls, which does not retain the crystal violet iodine complex. Instead, their outer membranes contain lipopolysaccharides, LPs, which are complex molecules that consist of lipids and sugar. This outer membrane makes them more resistant to certain types of antibiotics and disinfectants, and also gives them the ability to cause more severe. Gram-positive bacteria tend to be more susceptible to penicillin and related antibiotics, which target the peptidoglycan in their cell wall. Gram-negative bacteria are more resistant to these antibiotics due to their outer membrane, but are often susceptible to other types of antibiotics that can penetrate the. In summary, the main differences between gram-positive and gram-negative bacteria are their cell wall structure, staining characteristics, and susceptibility to antibiotics. The gram staining technique is a powerful tool that is widely used in microbiology to identify and classify different types of bacteria based on their cell wall.